Welcome to this video guide on monoclonal antibodies. We will cover everything you need to know for your GCSE Biology exam, from how they are produced to their vital uses in modern medicine. This video is structured into 4 key sections. We will start with a basic recap of the immune system, move on to the production of monoclonal antibodies, explore their medical applications, and finish with essential exam tips. To understand monoclonal antibodies, first remember that when a pathogen enters the body, our immune system detects molecules on its surface called antigens. The system then mounts a specific, targeted response to destroy these invaders. An antigen acts as a unique identifier on the surface of a cell or pathogen. They are essential because they allow the immune system to recognize specific threats and produce antibodies that fit them perfectly, much like a key into a lock. Antibodies are highly specific proteins produced by white blood cells called lymphocytes. Each antibody has a unique binding site that matches only one type of antigen, allowing for a rapid and powerful response when an infection is detected. While a natural immune response creates a mixture of antibodies, monoclonal antibodies are identical copies produced from a single clone. This consistency makes them incredibly precise and valuable tools for medicine. The production process begins by injecting a mouse with a specific target antigen. The mouse's immune system recognizes this foreign substance and starts producing lymphocytes designed to make antibodies that complement that antigen. Next, the antibody-producing B lymphocytes are extracted from the mouse's spleen. However, there is a challenge. These lymphocytes cannot survive or divide indefinitely in a laboratory culture on their own. To solve the problem of cell survival, scientists fuse the extracted lymphocytes with tumor cells. Cancer cells are chosen specifically for their ability to divide rapidly and indefinitely in the lab. The resulting hybrid cell is called a hybridoma. It combines the best of both worlds: the lymphocyte's ability to produce specific antibodies and the tumor cell's capacity for rapid, unlimited cell division. This sequence shows the logical progression of the process: injecting the antigen, extracting the cells, fusing them, allowing them to divide, and finally harvesting the resulting monoclonal antibodies. Once established, hybridoma cells are grown in large populations. Because every cell is a clone, they all produce the same specific, consistent antibody, which is essential for medical reliability. Finally, the antibodies are harvested from the culture fluid. They are purified and prepared for use in various applications, ranging from diagnostic testing to targeted therapeutic treatments. In summary, remember these 4 steps for your exam: inject the mouse, extract the lymphocytes, fuse them with tumor cells, and grow the hybridomas to harvest the antibodies. Monoclonal antibodies have truly transformed modern medicine and diagnostics. Thank you for watching this guide, and good luck with your upcoming exam. Monoclonal antibodies have a wide range of critical applications. The 3 primary uses you should know for your exam are pregnancy testing, measuring hormone levels, and identifying pathogens or delivering drugs. One of the most common applications is the home pregnancy test. These tests detect a specific hormone called HCG, which is produced by a developing embryo and excreted in urine shortly after conception. The test works through a simple process. When urine is applied, any HCG present binds to monoclonal antibodies on the test strip. This binding interaction triggers a visible color change, indicating a positive result. Monoclonal antibodies are also vital for measuring hormone levels in blood and urine samples. Because hormones exist in tiny quantities, their high specificity allows for precise clinical measurements without interference. In labs, they are also used to identify specific pathogens. By designing antibodies that bind to a unique antigen on the surface of a virus or bacterium, scientists can confirm the presence of an infection. This detection process involves taking a sample, adding the specific antibodies, observing the binding, and then detecting the signal. If the target pathogen is present, the antibodies will attach to it, revealing the infection. This technology extends beyond pathogens to cancer detection. Certain cancers produce unique antigens that enter the bloodstream. Using antibodies to bind these markers allows for earlier and more accurate disease diagnosis. Perhaps the most significant medical advancement is using these antibodies for targeted drug delivery. Often called magic bullets, they transport therapeutic drugs directly to cancer cells while sparing healthy tissue. The antibody acts as a homing device. Since it only binds to antigens on the target cell, the medicine is delivered precisely where it's needed, minimizing the damage caused by systemic treatments like chemotherapy. This targeted approach is a major improvement over traditional chemotherapy, which affects all rapidly dividing cells. By specifically attacking cancer cells, monoclonal antibody therapy results in fewer, less severe side effects for patients. The core principle underlying all these applications is simple: one antibody, one antigen. This high degree of specificity is what makes them so effective. Think of this as a lock and key mechanism. Because an antibody is shaped to fit only one specific antigen, it will not bind to other molecules, regardless of their appearance. This ensures accuracy in both testing and treatment. In short, monoclonal antibodies are essential in modern medicine. They serve critical roles in diagnostics, targeted therapy, for oncology, and fundamental research to further our understanding of complex biological systems. Ongoing research is constantly improving their effectiveness. Scientists are now developing fully human antibodies to reduce immune reactions and expanding their use to treat autoimmune conditions and even more types of cancer. Finally, be aware of the limitations. Early versions were mouse-derived and could trigger immune responses. Additionally, their high cost and the complexity of their production currently limit their availability in some healthcare settings. Let's look at a real-world example: cancer treatment. Herceptin is a well-known monoclonal antibody drug that targets a specific protein called HER2, which is found on certain breast cancer cells. By binding to HER2, Herceptin blocks signals that cause cancer cells to grow, effectively slowing or stopping tumor growth. Another common application is in COVID-19 testing. Lateral flow tests use monoclonal antibodies specific to SARS-CoV-2 antigens. If the virus is present in a sample, the antibodies bind to the viral antigens, creating a visible line. This works on the exact same principle as a pregnancy test. It is important to distinguish between monoclonal antibodies and natural antibodies. While your body produces a variety of antibodies to fight a broad infection, monoclonal antibodies are identical clones designed to target one single specific antigen. To excel in your exam, make sure you understand these key terms. An antigen is a surface molecule that triggers an immune response, while an antibody is the protein that binds to it. Lymphocytes produce these, and a hybridoma is the cell created for mass production. Familiarize yourself with these additional terms as well. Monoclonal means from one clone. A tumor cell provides the ability to divide indefinitely. HCG is the pregnancy hormone, and targeted therapy describes using antibodies to deliver drugs directly to diseased cells. When it comes to exam technique, remember that examiners are looking for process understanding and application. Don't just memorize the steps. Be prepared to explain why each step is necessary and how the unique properties of hybridoma cells are utilized. The golden rule for your exam is this: monoclonal antibodies bind to one specific antigen only. Whether the question is about diagnostic tests, cancer treatment, or lab production, always explain your answer by referencing this vital property of specificity. Avoid common pitfalls. Don't confuse antigens, which are on the pathogen, with antibodies, which are made by the body. Also, never forget to include the word specific when describing binding, and always explain the dual role of the hybridoma cell. Test your knowledge with these quickfire questions. Define a monoclonal antibody, explain why tumor cells are used in production, and name 2 medical uses of these antibodies. Check your answers against the provided notes to ensure you are exam ready. To wrap up, remember, monoclonal antibodies are specific, produced using the hybridoma method, used in both diagnostics and treatment, and represent an evolving field of medical research. Good luck with your studies.